So my name is Quabina Hopkins, and I'm going to be doing a presentation on the article titled Dynamic Reconfiguration of Structural and Functional Connectivity Across Core Neurocognitive Brain Networks with Development. And so early on, the article talks about sort of a little bit about brain development and how as we move from childhood to adulthood, the brain structure anatomically changes also, as well as the functionality of the brain. And so as these changes start to happen and the brain starts to mature, we begin to sort of get into high level cognitive abilities, such as, in the areas of working memory and attention. And these sophisticated abilities sort of rely on the dynamic interactions within and between three main large-scale neurocognitive networks. And so these three networks are the central executive network, the default mode network, and the salient network. And so the central executive network is mainly it helps govern maintenance and manipulation of information and decision-making in the context of goal-directed behavior. And we're going to be talking about two nodes specifically within each of these networks. And the nodes within the central executive network that we're going to be talking about are the dorsolateral prefrontal cortex and the posterior parietal cortex. And so when we move to the default mode network, the nodes that we're going to be talking about in this network are the ventral medial prefrontal cortex and the posterior cingulate cortex. And the default mode network helps sort of govern internally oriented and social cognition. And then finally, the salience network is the nodes we're interested in this network are the frontal insular cortex and the anterior cingulate cortex. And so the main job of this network is to govern attention, as well as interoceptive and affective processes. And so the article sort of said that this network actually is sort of could dynamically sort of control and choose which external stimuli we sort of give our attention to. And so the authors are seeking to fill the gap in literature. There have been previous network analysis and structural imaging studies that have helped us understand the dynamic maturation process of the brain. But the current authors want to better understand how structural changes impact the functional brain maturation. And so identifying changes in functional and structural connectivity is necessary to understand the dynamic reconfiguration of the brain as we develop from childhood to adulthood. And so the previously two nodes that I mentioned will be the main areas of interest within these networks. And the RFIC node specifically, which is within the salient network, has been shown to sort of initiate control signals that enable switching between the central executive network and the default mode network in response to cognitive demands. That's what I was talking about earlier with the salient network, able to sort of see see external stimuli and decide what we want to do with it. And so sort of the RFIC node acts as a causal flow outflow hub or coordinates other large-scale brain networks. I mean, in these brain networks, these hubs allow for increased flow of information between distant nodes. And so a causal outflow hub is a region that has a high number of causal outflow connections and a low number of causal inflow connections, as assessed by the Granger causality analysis. And so this GCA analysis is actually pretty helpful, to move beyond the identification of regional activations toward the characterization of functional circuits that sort of control, cognition, our perception of things, stuff like that. This GCA analysis sort of is a method that allows us to identify functional or causal interactions from time series data. So this GCA is able to help predict the effects of a lot of these things. And so that's why it's such a good technique to sort of use in this situation, that it helps identify the causal, the functional interactions. And so, basically, there are some gaps in the literature and less is known about the neural systems that support the development of these control processes. And so the authors are going to examine developmental changes in brain network interactions using multimodal imaging, combining functional and effective connectivity of intrinsic fMRI and DTI tractography. And so, DTI, diffusion tensor imaging studies, have actually shown that anisotropy increases and overall diffusion decreases with age. That's what previous studies have shown. And so this study is going to be sort of sort of looking at things from a little different perspective. And the author sort of hypothesized that they would find differences in causal outflow from network nodes between children and adults, with more dramatic changes in development detected in the RFIC node that we discussed earlier. And these changes would be further reflected in the structural connectivity measures. And one thing is important to note that the primary aim of the current study is not simply to link the statistical relations between functional and structural connectivity across large numbers of brain regions of interest. It's to sort of examine the developmental differences in the configuration and connectivity of these core neurocognitive networks. So right here is the first figure of that the article shows. And so the authors sort of computed instantaneous particle correlations between pairs of regions of interest to assess the developmental effects on the strength of functional coupling between the network nodes. And so partial correlation was a good statistical technique to use in this study, because not only does it measure the degree of association between two regions of the brain, but it's able to control for the effects of other brain regions that might impact the measurements you see. And so that's why partial correlation was pretty useful throughout this study. And so as you look at the figure, the nodes are sort of color-coded. The nodes and color-coded regions sort of were identified from ICA resting state fMRI data. And so it shows the salient network nodes in blue and the regions in blue, and then the central executive network in green. And then the default mode network it shows the yellow. And so it's just sort of showing the location and color coding them where they are. And so this slide sort of shows a multivariate GCA was used to examine the causal interaction between the 6 network nodes. And so we already talked about GCA and how it's good in examining these causal interactions. So the figure to the right sort of examines the functional connectivity between the nodes of the neurocognitive networks. And so as we look at it, the authors found that the RFIC connectivity with the ACC node, with ACC, DLPFC, and PCC was stronger in adults compared with children. And so when you look closer at part A, PCC and VMPFC connectivity were stronger in adults. And then when you look at part B, it's sort of examining the left hemisphere counterparts of these nodes. And the only link to have found a group difference was the left FIC ACC connection. And so these findings sort of help authors understand that both within network and between network connectivity patterns are stronger in adults. And so when we look at this figure, it's sort of similar to the previous one, but this figure examines the differences in strength of causal influence. In this figure, this figure examines the differences in strength of causal influence between the adult and the child group. And so looking again at part A, it shows how the interaction from the RFIC to the ACC and from the RFIC to the RDLPFC was a lot greater in adults than children. And the IFIC ACC connection was also shown to be greater in adults. And so this actually was pretty important, because it helped the authors understand that IFIC is not as strong a driver of network dynamics as the RFIC node is. And so this is when they start to sort of understand things a little bit better and start to specifically, look at the interactions, because a lot of things, previous studies have had a general outlook, but this study was looking specifically at which nodes, which networks, and which small specific interactions were impacting this maturation process, and being the primary driver of these neurocognitive networks and their interactions, stuff like that. And so look at this as we look at this slide, like similar to the previous one, but it's sort of looking at a matched data set, and so a group of adult participants and child participants who were the same, did not differ in mean age, distribution, or IQ from the original sample, were matched, and they used the same statistical analyses that the original data set went through. And so this figure shows that the differences in effective connectivity observed in the original sample were replicated with the matched data set, with only one additional group difference observed. And so adults were greater than children in the VMPFC OCC causal influence in part B of the figure. And so as we look at this figure, the authors calculated the net outflow of causal interactions in both groups to see if the previous findings that suggested that RFIC node is a causal outflow hub were actually true. And so one thing that this study actually expanded on was they were seeing for the first time whether this holds true in children. And so when you look at the figure to the right, it shows the result of a network analysis computing the out-in degree for each node of the 3 neurocognitive networks. And so the results showed that in both children and adults, the RFIC had significantly higher net causal outflow than the ACC, the PCC, the VNPFC, the RDLPFC, and the RPPC. And so right here, they're really sort of zoning in on the RFIC node. And this node in this area having a significantly higher net causal outflow, which is, which we previously said sort of is big in sort of controlling these interactions and affecting these interactions and stuff like that. And so they're zoning in on just a specific area and a specific part of the brain that has this massive impact. And so previously we sort of said that the primary aim was to examine the developmental differences in the configuration and connectivity of core networks. And so, now we're looking at the structural connectivity. And so one important thing to sort of outline here is that structural connectivity refers to the anatomical organization of the brain by means of fiber tracks, whereas effective connectivity is defined as the influence that a node exerts over another under a network model of causal dynamics. And so using deterministic tractography, they found that adults had greater structural connectivity compared to children, a finding that was sort of echoed in previous studies. And so when you look at this figure, it shows the structural connectivity between the salient network nodes, RFIC and ACC. And so you have in the white, when you look at the figure, the white part is the tracks that are connecting these two nodes. And so in part A and B, you can see the structure of the brain and comparing the two between the child and the adult. But then when we look, you have part C and D. And so the figure shows that the children were shown to have lower mean fiber density in the RFIC ACC tracks when compared to adults. And that's in part B, where you see that the fiber density is lower in children than adults. And so and then also an important part of this is part C, where the fractional anisotropy measure showed a similar trend to that, but there were no significant differences between the group. And so FA, the fractional anisotropy, sort of describes the degree of anisotropy of a diffusion process. A lower value in this means that diffusion is isotropic. It's sort of unrestricted in all directions, but a higher value in this means that diffusion occurs only along one axis and is fully restricted along all other directions. And so that's a little background information on sort of what these measures mean and sort of how they affect the brain a little bit. And so this figure is sort of is sort of similar to the first one that's measuring this the connection, the link between these two nodes. And so this figure shows the structural connectivity between the RFIC ACC link, but it's in 8 individuals. And so it has 4 children and 4 adults in this figure. And so DTI tractography sort of showed that the tracks along the uncinate fasciculus can be identified readily in individual subjects. And so that's what this sort of is showing, that in individual subjects, you can see that each individual, all 8 of them, you can see the areas in the connection between the tracks and stuff like that. And so you see the 2 nodes and you see the white tracks sort of connecting them. So this one is sort of similar to the previous one, but it's looking at a different connection. And so this figure shows the tracks between the RFIC and the RDLPFC. And so the figure shows that the mean fiber density showed a strong trend toward being lower in children compared to adults. And again, the FA measure shows, but differently than this one, the FA measure shows significant group differences. And so, but once again, you see that the fiber density is higher in adults. You see the FA is higher in adults. And so, what they're sort of seeing is that, adults have, a greater structural connectivity, a greater functional connectivity. And so as the brain matures, these connections get stronger, these interactions get more dynamic and stuff like that. And so it increases our ability to perform these higher cognitive abilities that underpin these structures in these brain areas. And so as we move on to the next figure that they sort of discuss, this figure shows the structural connectivity between the nodes of the salience network and the central executive network again. And so it's looking at that same link from last time, but it's in 8 individuals. And so it's showing the 4 children and four adults once again. And so what these figures have sort of been showing, these past couple of figures have been showing, is that pathways showing both reduced functional and effective connectivity in children compared with adults also showed absent or reduced structural connectivity as measured with DTI, the diffusion tensor imaging. And so that's important because it shows the connection between the functional and effective connectivity and the structural connectivity. And that's sort of what they want to better understand, how these two sort of interact and how they move and how they sort of interact and mature together and impact one another. And so to explore relationships between functional and structural connectivity a little more, the authors computed correlations between between functional and structural connectivity in both groups for the RFIC-ACC link and the RFIC-RDL-PFC links, as shown in the figure to the right. And so once again, you see on the y-axis, you see the partial correlation, and then on the x-axis, you see F8 fractional anastropy, and so anisotropy. And so the RFIC RDL-PFC link had a significant correlation between the functional and structural connectivity in adults. However, there were no significant correlations seen for the other link in either group. And so this is just sort of better understanding the relationship between the functional and structural connectivity, and better understanding, which group has stronger links, stronger interactions, and which group has a stronger correlation and stuff like that. And so adults, have been consistently, as the study has been going on, adults have been consistently sort of having stronger links, having stronger interactions, having more dynamic interactions, having these sort of increased fiber density, increased FA and stuff like this. And so it's just showing the maturation process from a childhood to adulthood, and how adult brain networks are sort of at a higher level in a lot of these sort of areas that they were looking at here. And so as we sort of, as this article as the authors concluded, the basic thing they did throughout this study was examine the developmental changes of the core brain networks using various functional, effective, and structural connectivity measures. And so their findings were able to confirm the causal influence of RFIC on other brain networks areas become stronger as we develop from childhood to adulthood. Children also show lower structural connectivity along major links than adults. So so the adults sort of were, at a higher level, functionally, structurally, and with effective and structural connectivity measures. And so they also showed how the RFIC connectivity had the most dramatic developmental effects. And so some of these studies previously that sort of outlined this as a potential, area of interest, the authors were able to examine it specifically using their measures. Instead of some previous measures that had been more broad, this study was very specific and able to pinpoint that the RFIC connectivity had this major impact. And so in future, when looking at future studies, and looking at the importance of this RFIC node, the pathways are also important for understanding neurodevelopmental disorders involving disrupted connectivity, such as autism spectrum disorders. And so ASD participants have decreased functional connectivity between the RFIC and ACC nodes. And so basically the results of this study have suggested structural and functional maturation of the RFIC pathway is a critical component of the process by which human brain networks mature during development to support the complex, flexible cognitive processes in adulthood. The findings from the study are very important for understanding the development of increasingly sophisticated cognitive control abilities by this multi-network system, and able to point to a potential locus of dysfunction that may be targeted for additional investigation and studies of neurodevelopmental disorders involving the RFIC. And so the authors really were able to sort of outline this node, the RFIC node, and outline its effects and how crucial it is for the development of our brain, and for the development of also these other neurocognitive disorders that could affect people, such as the autism spectrum disorder. And so that is basically the main findings of the study and the main results in the potential future directions and implications of the findings of this study. And my name is Quabbin Hopkins, and this is the end of my presentation.